Hello, and welcome to lecture number 8 in Systems Biology of Aging. Aging here and there. Aging. You can sing with me. I think it's lecture number 8. Aging here and there. Aging everywhere. If you're a whale, you can live a century or two, but you won't make it to your second birthday as a shrew. You know? Yes, some critters are immortal in a fascinating way. And you may learn their secret if you concentrate today. Aging here and there. Aging everywhere. Aging everywhere. Aging. Hi everyone, let's take a nice big sigh of relief. Whoa! That was cool. Yeah, everyone took a sigh of relief. You missed the song. Should we do it again? Yes. Okay, welcome. Breath, song. I'll just do the chorus for you, okay? Just for you. I'll do the chorus, okay? Because it's part of our material, okay? Ready? If you're a bowed whale, you'll live a century or two, but you won't make it to your second birthday as a shrew. Right? There are immortal critters in a fascinating way, and you might learn their secret if you concentrate today. Get it? Animals here and there. Animals everywhere. All right, so we all love animals. It's a great topic. So just to review, so in the last 2 lectures we talked about, I turned into an exercise coach, exercise and longevity, and then nutrition and longevity. Remember? I just want to finish off because, what's your name? Dan? Dan asked me about sleep, right? So we have our 4 elephant legs of well-being, which is exercise, nutrition, social relationships or emotional well-being, and sleep. It's equally important. It's also very undervalued in our society. It's so undervalued that lazy people sleep. I'll sleep when I'm in the grave. You will sleep in the grave if you don't sleep. But people say instead of a nap, we call it a power nap because it has to be powerful, right, and effective. But you need 8 hours of sleep, or at least the opportunity for 8 hours of sleep. And less than that raises risk of all kind of age-related diseases. But and then there was a question of coffee, so I just want to do this little thing here about coffee. So sleep is governed by 2 forces, right? One force is the clock that we have, the circadian clock, that we get cues from sunlight, and it's really good 24-hour clock. Even if we're in a cave and there's no light, it still runs, but not 24 hours, a little bit different. It's called circadian, but with light it gets really entrained very well. And it's a wake-up signal. So this is a wake-up signal. For example, you get a surge of cortisol. You wake up. Some people, for me, it takes longer. Other people it takes longer. Right, so you wake up. Now, as you wake up and start behaving, there's also a sleep signal. That's a buildup of a chemical in your brain called adenosine. And it starts building up. So this is wake signal. Wake up. And this is the sleep signal. And then when we fall asleep, so let's say we fall asleep around here, it starts degrading. Right? So that's the normal. What does coffee do? It blocks the caffeine receptors. So it's like the caffeine is saying the sleep, so the adenosine receptors. Adenosine, it has receptors in the brain, and caffeine blocks them. So adenosine is screaming, go to sleep, but caffeine blocks it. but you still have that debt. Right? Now, what happens, for example, if you don't sleep, if you don't go to sleep? So you're supposed to go to sleep here, but you don't. You pull an all-nighter. You're very tired. This is how tired you are, right? The difference between sleep pressure and wake-up pressure. But do you know this phenomenon that after a while you get a second wind and you feel more awake? That's here. Second wind. And then after 48 hours, this has built up a lot, and now you're so tired, you have no chance. You're going to go sleep. So this is like high pressure to sleep. Now the coffee, you have to know, it has a half-life, and that differs between people. So it could be 6 hours, 8 hours. Some people are not so sensitive to coffee, but if it's 6 hours, then it goes, so when you drink it at noon, your coffee, your espresso, at 6 p.m. you have half an espresso in your body. So that should be important for you when to time your coffees and teas. And you said you're going to do a final project, right? Which is?
About our addiction to coffee.
Yeah.
And how to simulate a model.
Yeah, so if suppose you had an app that tells you what if you drink coffee at these times, what's going to happen to your sleep pressure. Also from your genome you can tell how sensitive you are. And this is also time to say about the final project. It's a perfect final project. So pair up and think about interesting questions you can deal with the tools in this. How many of you have an idea what you want to do in your final project? Okay, good. So how many of you have a partner to work with? Almost everyone. Okay, that's good. If you don't have a partner, I can help. Ben can help you. All right. Okay, so 8 hours of sleep opportunity. And of course, you keep the word dark, no gadgets. But there's one interesting thing that happens when you sleep, the circadian rhythm. One of the circadian things is your core body temperature drops at night. It turns out that if you feel cool, you fall asleep better. Do you know that? So, for example, taking a warm shower before you go to sleep, you actually feel cool after you go out of the shower, and it helps you sleep. So there's interesting thing, and I want to recommend the book Why We Sleep, which is written by sleep scientists. It's real science, but it's science communication. It has all this information and can tell you there are some depressing chapters about what happens to you if you don't sleep enough, because the brain, for example, cleans itself up at night. The role of deep sleep and REM sleep in consolidating memories and your emotional health. If you don't sleep enough, you'll not sleeping a good night. You try to do an exam, it's like being legally drunk, for example, in terms of your focus. It's amazing how important and undervalued, almost a taboo subject in our culture. But okay, so we looked at last time. We looked at nutrition. We looked at the weight set point model, eating versus fat, energy line, appetite line, and how if you exercise, you eat more and you lose weight. If you grow old, basically, you gain weight. If you use GLP-1 agonists, you lose weight. When you stop, you have an overshoot. We did all that analysis last time, and we also talked. So I want to now switch gears. What I want to do in the next 2 lectures is talk about the molecular aspects of what is the driver of aging in our body, molecular, biochemical, cellular, and approach it light for today, which is looking at different animals, what we can learn. Some live long, like the whale. Some can't make it to their 2nd birthday, like the shrew. What we can learn from these differences. And then next lecture will be about genetics of aging. What we can learn from centenarians, people who live to 100, and what we can learn from genetic screens, and really get to like what we think are candidates for the biological drivers of aging, where you can intervene. So it's these two. That's why I want to talk about comparative aging, or aging across the tree of life, the evolution of aging today. You ready? Okay. Before I do that, maybe there's a question about the nutrition or exercise lectures that's in your mind, or sleep. Yeah.
What are the effects of iron or adrenaline on these ideas?
The effects of iron or adrenaline on sleep.
Iron deficiency
Iron deficiency.
or adrenaline when we have high adrenaline, we don't want to sleep.
Oh, high adrenaline.
Yeah.
So adrenaline, I think, is one of those things that will help you wake up in the morning. But I don't know about iron. Do you know about iron? Sleep and iron. I don't know.
Makes people more tired. Right?
Yeah, anemia, lack of iron.
Yeah.
What? Because of oxygen?
No, it is.
You can't transport oxygen well enough? You think so? I don't know about
You
that.
generate less hemoglobin. But
You generate hemoglobin. Yeah, I don't know about that. But that might be a good final project. Iron and aging. You know, in aging, by the way, iron levels go up too much. In aging, for example. Why? What's going on there? Okay. All right, so animals. So from the time of the ancient Greeks, it was apparent that bigger animals live longer. You can actually find that in Aristotle. And then in the mid-20th century, there was some work that tried to plot mass versus maximum lifespan, longevity, max lifespan observed, let's say, in the zoo or something. And so if we put here, now I'm talking about mammals, for example. If I put here 1 gram, in 2 years, I have the shrew. And here is the mouse. And that other extreme, weighing in at 100 tons, which was 100 times 10 to the 6 grams, which 10 to the 8 grams, we have the bowhead whale, which lives 211 years is the maximum known. Okay? So the mammals, 35 million years ago, they had a common ancestor. And they evolved very large body size differences and longevities. Right? And then there's other animals in the middle. Like, do you know how much dogs live? Huh? Say 20. 20 for small dogs, 12, let's say. And then how much do they weigh? Huh? 10 kilos. Yeah. All right. And then we have other animals. Where is people? 100, yeah, let's say 120. 119. And how much do we weigh in grams? Let's say we're talking about orders of magnitude. 10 to the 5, 100 times 10 to the 3, 100 kilos, 10 to the 3. I do it like this, even though I'm only 90, but who's counting? All right, so it looks like there's a line here where L goes like mass to the 1/4. So that means root of a root of mass. So let's take, for example, if we have a 10 to the 8 whale, okay? 10 to the 8 whale lives 200 years. Then a 10 gram mouse, so 10 to the 8 grams to the power 1/4 will be 10 to the 2. And this is 200 years. And this is like wait. What?
The 2 in front is in order of
Yeah, right. So wait, I want to see. So if I go, what I want to say is the ratio between a mouse and a whale is 10 to the 8 to the power 1/4, which is 100. So 200 years versus 2 years. Right? And it's a really, really powerful idea, this thing called allometric law. And there is indeed a very good allometric law, which is your power, which is how much you can say energy per unit time, calories per unit per hour, whatever, versus mass. And that's goes like mass to power 3/4 or 2/3. There's a debate, but this means this is probably because of surface area. So small animals have much more surface area compared to their volume, and they need more energy to keep their heat. So this is the total power, and you can have mouse and whale, and therefore power per unit mass also goes to m to the minus 1/4. So people said, wow, a cell from a whale is 100 times less power than a cell from a mouse. Because this is total watts. This is watts per unit mass. It goes down with mass. Long-lived animals live slower in terms of their metabolism. So maybe it's metabolic rate that controls lifespan because of this association. Okay? So this was the theory of rate of living. Theory, which said that metabolic rate 1 over lifespan. So faster you live, the faster the candle that burns brightest burns fastest. This was Pearl. There's someone in the 1920s that had an influential book about that. And also another influential and nice thing about this is that it turns out that the number of heartbeats, the heart rate also goes like mass to the minus 1/4. By the way, mice have a heart rate of almost 1,000 beats per minute. So we have 60 beats per minute, right? Depends, can be 180 when you're running. Mice have almost 1,000. So that's thought to be said the limit for the smallest possible mammal is this thing. Because you can't do 1,000 beats per minute with your mouth. It's like much more than that. It's like, how did they do that? I don't know. But the heart rate times longevity, this is our total heartbeats per life. So it's very nice that you have, it was constant, it's about 3 billion. So it was very enticing to say, wow, we have a limited number of heartbeats. It's like romantic, right? It's very good, but it's wrong. Because, this law seems to be quite accurate, and I think all the mammals fall on it. But this law is not accurate, and the people who did it actually did cherry picking. That's to say, they left out some data because of the bats. Yeah. So the bats, like Brand's bat, few grams, 40 years. But that's only the bats. There's what's called naked mole rat, which lives underground, also about 40 years, et cetera, et cetera. So they have, the bats, they do have the predicted metabolic rate for species their size, but the number of heartbeats is much larger. So it's not the rate of living that determines lifespan. For example, there's many other reasons to say, but just to be careful of. So in all this course, we're looking at patterns and stuff like that. You can, of course, be misled, but it's very important to have good data. And there's questions.
This here doesn't apply to a single species, like so. if we think of like a number of human beings, right? Certain number of people have higher metabolism. Then by this theory, the lifespan of those people should be less, right?
Yeah, this theory does not apply within a species. We'll talk about that. Like large dogs live more or less than small dogs? Less. But large animals live more or less? More. It's the opposite. So we'll talk about that. Very interesting. Because we want to explore, in the end, what biologically sets the lifespan. And evolutionarily. So what is going on? So here is work by, I mentioned this, I know, in the first lecture, but I want to go into it more in depth. All right, so I'm going to erase all this stuff. By the way, it's very important to have wrong theories. If you don't try, you don't succeed. And you can't be frozen by the fear of errors. So this is work we did with this PhD student, Pablo. And he plotted, there's some interesting talking, which means you're probably really interested, right? And you're really interested and you have a question maybe.
What happens with females in other species? Do they also live longer?
So in many species, females live longer, but not in all species. Yeah.
You said cherry picking. Do you know if they did it by mistake or intention?
I don't know. I don't know the history. But it's seductive, though, to have power laws. So this one, Kleber law, checks out, as far as I know. Also can be extrapolated to down to bacteria. It's not clear if the power law exactly, because it's hard to tell, but something like that. But all right. So, but here it's really different from a line. So it's basically looks like a triangle. We called it the mass longevity triangle. Data is from NH database, which collects life history facts like mass, longevity, gestation time, pregnancy, birth weight, litters per clutch, so how many babies you make in one, things like that, for different mammals. And also for birds, you can find, which are, both of these are the species that keep the temperature. They're called endotherms, as opposed to turtles, which can live a long time, but they have the temperature of the environment. They can't keep their temperature. They have a different, very hard to pronounce word for what they are. Does anybody know? You're all molecular biologists or physicists, right? No zoologists here? Poikilotherms. You hear about this?
Would it be endotherm and ectotherm?
Ectotherm. I don't think it's ectotherm, but all right. So here is the shrew. Here is the bat, and here is the whale. Here is the African elephant. 6 tons, 65 years. Here is the human. Here are some primates. Here's the cow, dog, bear. Here's the bats. Bats is a big clade. There's some squirrels. There's the naked mole rat. And it's kind of full. All right. So when we look at biological data and we see that it fits inside a polygon or a polytope like a tetrahedron or whatever, we can invoke evolutionary theory of trade-offs. So the evolutionary theory of trade-offs says that there's no animal that can swim like a dolphin, run like a cheetah, and fly like an eagle. Why? Because you can't dance at 2 weddings with one body. You can't have it all. So when you have multiple tasks that you need to embody with one phenotype, you have to have trade-offs. But it turns out that these trade-offs have a very special geometric shape in the space of characteristics. And that shape can even tell you how many tasks you're trading off. Because a lot of times we look at nature and we say, oh, this beak needs to eat insects and crush. But how many tasks does it have? That's our mind imposing. But if we look at the number of vertices in the shape filled out by the data, we can know how many tasks there are. Let me demonstrate. So imagine that you have Darwin's finch, like you have a finch beak. Okay, in the beak, there can be different traits, like the width, the depth, etc. Okay, size, length, whatever. Now, beak can have different tasks, right? If you want to eat small seeds or big seeds, or nectar from a flower. So for large seeds, you need to have a plier. And for this, you need to have a pincer, right? But you can't be the same with one beak. There's no way for one beak to be a plier and a pincer. So what happens? So what you do is you imagine, let's imagine that there's, first of all, let's imagine that there's only 2 tasks in the world. Just imagine. And I plot the optimal design for task 1. And this is the optimal design for task 2. And we call them archetypes. They're archetypal. And you see they're different. That's why there has to be a trade-off. You can't do both with one beak. And now I'm going to ask, what about multitasking birds? They want to do both. Okay? So I'm going to say the performance at task 1 decays or goes down with distance from the archetype. Right? The farther I am, the worse I am in this task. And the performance at task 2 decays the farther I am away from this archetype. Right? Because they're the best. Now, if we look at the line between these 2 archetypes, yeah, I can guarantee you that there's a design that's closer to both archetypes than this. In fact, the line is a set of points which are closer to those both archetypes than any other point. So since there's a point here that's closer than here, I can erase this point. Natural selection will not choose this point because this is better at both tasks. And I end up with this line is the expected suite of variation. Yeah.
But that section might choose like that point on the bottom right if they're only looking at that one's task 1, right? Like the task 1,
if you have only task 1, you'll go here.
Yeah. But what if it's looking at the 1 axis and like that's what's really important?
If there's some constraint, then if some constraint we don't know about, then you'll hit a wall. Right? If you can't go above this point, you'll hit a wall. But if you can, you'll get there. That's the theory. If natural selection is working at it, and there's no noise, and there's no errors, and there's no speciation and migration, and okay. So if we see data, we take 2 traits, like mass and longevity, right? Whatever. And we see data on a line, we can imagine that the ends of the lines are archetypes. Okay? There are a lot of things like that. What if we had 3 tasks? And in reality, 3 tasks. So you have 3 tasks. What's the optimal place to be? Inside the triangle. So the triangle, if I have a point inside the triangle, there's a point inside the triangle that's closer to all 3. So I can erase this point. That's called Pareto optimality. If you have a design that's better than another design, if there's 2 designs and one is better in all ways than the other one, you can erase it. Okay. Where you are depends on the niche. So when you look at Darwin's finches, they actually fall. There's like 6 species, and they fall in a triangle. This one eats cactus. This one eats large seeds. And this one's eats small seeds. And these are generalists. These ones are specialists. These ones in the middle of a triangle are generalists. See where I'm going? So when we see a triangle, we think there could be 3 tasks. And then we look at the organisms closest to them and ask, what do they do? And we can build a hypothesis what those tasks are. We can discover tasks based on geometry. What if I have 4 tasks? In order to see it, I need to measure 3 different traits and 4 different. So the 4 optima define 4 points in space, and you predict a tetrahedron. And in fact, this is very powerful. Like, if you look at the single-cell gene expression, let's say of liver cells, they have many tasks. You plot them on principal components of gene expression, and you see they fall in a tetrahedron. And there are archetypal cells, and they do things like one does detoxification, gluconeogenesis, albumin production, etc. Aha. Right, so here you would say there's 4. But of course, it's a statistical question how you say, is it significant inside a polytope, how many? There's algorithms for all that. That's called Pareto task inference. Because you infer the tasks. And this is the paper. All right, so when we look at this data, we're primed to say maybe there's 3 archetypal strategies here. What are they? I just want to say in ecology there's a classic, what's called RK strategy. Live fast, die young, leave a good-looking corpse. Mice. 0 predation, you never starve, you can live long, have 1 baby at a time, grow slow. Whales. Okay, so that's a continuum. So we know, when we look at it, we see this, we look at the animals close to here, and we see they have many babies. How many babies do mice have per shegel? Who works with mice here? How many?
10.
Huh?
10.
10, okay. And then they have very, very... okay, there's lots of like optimal things near this archetype. These guys have 1 baby, well spaced. But these ones have very small brain to body mass. So they can be big and stupid because they're not stupid because they have big brains, but their resources aren't going especially to brain. They're very smart, dolphins. Okay. What about the 3rd archetype? So it's well known in evolutionary theory that what drives the lifespan is extrinsic mortality, which is to say how fast you starve or predated. So like if you're a mouse and you live 1 year, you don't need to invest in repair and slow growth, because which can take you to 10 years, because you're just going to be killed in 1 year. So instead you invest in fast growth. And indeed mice have very poor repair, like poor DNA repair. Humans have much better. So what about the bats? These species live in niches which are called protected niches. For instance, bats can fly, right? So this is the 3rd archetype here is the protected niche. And they have protected niche. They have large brain per mass. By the way, primates, naked mole rat also close to there. Naked mole rat lives underground, so protected niche. They have longest time between births because they teach their kids. That's a little bit like people. So some bats carry their baby on their back to teach them where the fruit tree is 20 kilometers away, even though they pass some fruit trees on the way. So it's very interesting, this cognitive niche or near the squirrels, flying squirrels or boreal creatures on the trees, all these kind of things. Also, birds that can fly are up there, and birds that can't fly are down there where the cows are. So like, and animals, these animals here have what's called a large longevity quotient, which means that they live longer than what they should according to their mass. Okay? So they're very interesting to study when you do molecular studies, right? What's the difference between them and mice, let's say? Because you can tease apart the mass effect. If you compare mice to whales, there's mass differences, but if you compare mice to bats, naked mole rats, you tease out some of the effects. Also, you want to be maybe in the same family or order. Like if you're in the rodent family, you want to compare rodents to rodents. And now, this is when you plot two traits. What happens when you plot other traits? So there's a lot of missing data, but there's one more trait you can work with to do a three-dimensional. And then there are hints, actually, in this data of a tetrahedron. This is the triangle. And above this point, there's another archetype. And close to this archetype are panda. Oh, it's the archetype of cuteness. Bears, lions, like large carnivores. They have middle mass and middle lifespan. They have they have a big ratio of mother to birth weight for some reason. And that requires more study. But the better the data you have, you can resolve more archetypes. So this is like about the comparative facts of, let's say, or ecology or ways of thinking about differences between male and species. Where are we? 1/4 law, mass longevity triangle, archetypes, Pareto, tetrahedron. Oh, and now we come to dogs. So there was a comment that within a species, we call this archetype X because we don't know what it is. What are you photographing?
I want not a view of the like hand of tetrahedron just because
Oh,
I had a joke.
okay, like archetype of cuteness. No offense to other animals.
Where's the koala?
Koala? Yeah, so marsupials are also in here. Marsupial is a great test. Maybe this only comes from phylogenetic relatedness. Maybe animals close on that family tree are there just because their parents were there. So in order to see that, you see that mice and naked mole rats are really far away. And some relatives of whales are here, etc., etc. So phylogenetics doesn't explain most of this variation. So another way to test it is marsupials, another experiment in mammals, but they have a different system. And they have a similar triangle. So there's like analogs for bats, whales, and rodents for the Australian. What is your question?
So what you're saying is that those mice die out of age at the age of 2, like out of aging.
Yeah, so the mice, do they die of aging at the age of 2? So if you look at mice in the lab conditions, they reach about 2.5 years, depends on the strain. And they look very old. They have kyphosis, which means bent back, cataracts, gray fur, hair loss. A lot of cancer, lymphomas go up, kidney disease, need I go on? They really look old. Frailty.
And they die from that.
And they die from that. Primarily from cancer, but also other reasons.
But so those mechanisms essentially got tested to see exactly why do they age.
Yeah, why would they age so fast? See, that's the point. Why do mice age so fast and humans age so slow? That is the question, right? Yeah, that's why we're here, right? So tell us, right?
I'm asking you.
Yeah, you wanted me to tell you. Yes, I want to tell you. But in my own pace. We're coming to it. And that's these 2 lectures. I wanted to know what the fundamental biochemical mechanism is of aging and how it's tweaked, etc. So I'm so glad you're with me on the same web page. Okay. You know, when you're in acting school, you learn ways to like prolong the drama. Okay. About dogs, yeah, there was a question. Yeah,
So you said that the mass to
mass to what? Surface area. Surface area of an organism. Yeah. By the way, it's good to say in Hebrew. I'll just translate. Yeah. You can ask in Hebrew if you want.
With the mass, like the
Yeah.
small organisms live last.
Surface area. You're asking. So surface area is like meters squared. By volume, which generates the energy, is meters cubed. So surface area goes like mass to the cube, 2/3 law. If we were exactly scaling, but we're not exactly scaling. That is to say, if we increase the animal, it's not that the shape is. We're not spherical cows, so it could be.
It wasn't the whole
So what's the question? Yeah, I know. Sorry. Surface area of animals.
You said that this relates to the fact that small animals live shorter life.
So this relates to short animals, to small animals live shorter life.
So how does it work within species?
Within a species, right.
Smaller.
So I wanted to correct myself. In the past, people thought that this is the reason that small animals live shorter lives, or that was one thinking, because they have a faster metabolism to heat up. Of course, cold-blooded creatures don't need to do that, but still have faster metabolism, but okay. And the faster metabolism means they burn out faster. But then the bats destroyed that notion because they live long and have fast metabolism. I mean, they have the correct power according to the mass, but much longer life. Did I explain myself? Yeah, so this is one example of disproving a theory or... I mean, of course, metabolic rate does affect the rate of house production and toxicity per house, but it's more elaborate than that. It's not that alone that... So I just want to guide you through history of ideas in this field. Yeah. Toma.
What's the extra axis of the tetrahedron? The additional.
This extra axis? So you can look in Pablo's paper, but it has to do with birth divided by mom weight. So the ratio of the newborn related to the mom. Because that's the third most thing with the most data. But maybe now, this was 2016, there must be more data. It's time to revisit and get more information. By the way, when we did that paper, I still didn't know we were going to study aging. I just thought it was a cool way to demonstrate triangles. Now, I think maybe that's why I started. I don't know. Maybe. Yeah. Well, listen, I love your questions. I also want to go forward. I have an inner tension. So let me take one last question.
I was curious, like, also it's just like the missing data, but maybe also things like environment and like temperatures where the animals live, and also like say dietary patterns. Like, is there information on that?
Yeah, the information about environment, dietary patterns, of course, will affect the maximum lifespan, which itself is not a perfect measure. I want to say that cold-blooded creatures generally live longer at low temperatures, and that really does determine their metabolic rate. So maybe there is something about metabolic rate, but we'll discuss that. Okay, dogs. So just to say about dogs, so the dog is here, but within the dog clade, smaller dogs live longer. And in the lecture notes, I prepared for you survival curves for terriers, mastiffs, and if you like dogs, pure breeds. And they change both in their median lifespan, but also in their steepness, so we can use the SR model to figure out what's going on. One major thing that happened was that, so what I want to say here, breeding, because they all come from the wolf, right? These are all breeds, they're not different species. They can mate. Breeding tunes lifespan differently than natural selection. Just different knobs. And one of the knobs that was mutated in the dog evolution was this protein we talked about before, IGF-1, which is a signal for growth. And mutations in IGF-1 that lower it reduce size and increase lifespan. High IGF-1, maybe we'll talk about molecules. In studies, and there's many studies that compare what's different about these animals and these animals and these animals, and centenarians, etc. One of the commonalities is that the longer lived species have lower IGF-1 pathway. So this, what does this do? This is, in one of its functions is, as I told you, a knob that senses the nutritional environment and decides if the world has nutrition, I should reproduce and grow, and if the world is starving, I should repair myself and hold up until there's food. So it's like and all species have that. This IGF-1, mTOR, which is the protein computer inside the cell that tells make more proteins, tells the ribosome to make more proteins, and stop repair. So if I lower this, I make less protein and I enhance repair. Both of them pro-life, because proteins made cause aggregate problems. Repair, of course, is pro-long life. So this is a common thread. And this was mutated in the breeding of dogs. So Great Danes have a lot of IGF-1. They grow fast, fast, fast. They die young, young, young. But there's not the only reason. We know that from the shape of the survival curves, there's more reasons. It's very interesting. Another maybe project if you're a dog lover. So actually there's now, in the last 6 months, veterinarians in America can buy longevity pills for pet dogs, especially large breeds. They work on IGF-1 receptor antagonism, and the company says it will enhance the lifespan on average by 6 months. So that field of companion dogs is actually a field where longevity clinical research is the most advanced. There's projects where dogs and their owners do clinical trials on things like rapamycin, and there's a couple of companies that sell products already that are like scientifically, I don't know how good they are, but so veterinarians are ahead. Isn't that interesting? Yeah, also for cats.
They expand lifestyle.
Too many questions. Right. I didn't want to offend you. Keep your question. All right, so where are we? Dogs. Look, we only made it past the dogs. I knew the dogs would steal the show. The small dogs and the big dogs. Age, survival, the big dogs, the small dogs, the medium dogs, the big dogs, the small, thank you, small dogs, actually the medium dogs. The giant dogs have a lot of childhood, like young mortality. And some breeds that really have a lot of mortality because they can't eat, they can't breathe. Huh? All right. Let's just do a lecture about dogs. Who has a dog? Who has a cat? Who doesn't have a pet? Okay. I have both. Which is what? Pets? Cuteness? Another leg of the elephant? Yeah, but it's part of the emotional health, right? If you take care of something, you fall in love with it. Okay. But I'm going on a tangent, so let's clear up the board here. After the break, we're going to talk about immortal animals. So we're going to take a break now. We're going to talk about immortal animals, and we'll talk about the molecular differences between long-lived and short. So right now it's 3:06. We'll get back at 3:18. Okay. Yeah, I was asked some questions. I just want to. I was asked some questions. So one question has to do with how do we know that evolutionary optimization is over? I mean, maybe if we have more time, things can move. So it's possible. We did one study to explore that with a group of animals called ammonites. Ammonites are these shells that look like this. You might have seen fossils. And they're used in the past to date rocks, so there's a lot of data about them. And when you look at their spiral shapes and you plot two different aspects of the spiral shape, you get the ammonites fall in a triangle, and then you can deduce what their tasks are. One is economy, minimum material in the shell to get the maximum volume. Another one is hydrodynamic speed. And the third one is rapid growth. So you have specialists and generals. Now, because it's 300 million years, there was a mass extinction in which only two genuses survived. And after 20 million years, they refilled the same triangle. Then there was another mass extinction in which one survived, and within 20 million years refilled the same triangle. And then with the dinosaurs, another mass extinction, and today we only have one genus called Nautilus, probably because other niches were taken by marine mammals. So that tells us that the archetypes are constant over geological timescales, at least for ammonites. You know, species time. Another piece of evidence is Darwin's finches. Why did Darwin get so excited about Darwin's finches? They're on an island. They were similar to a finch in South America that came. In South America, the niches were filled by other birds, but now this finch is alone on the island. And it says, oh, look, there's some cactus. So the finch has some genetic variation in their beaks, and the ones with a smaller beak can make a living on the cactus. And then there was a speciation. It took 1 million years to form 6 new species from this one bird that migrated, one bird population. So that's the speed of this thing. But maybe it's not over, right? Another million years, maybe it'll expand, who knows? But okay, and of course that's with the constraints. If the beak would be made of titanium, it'll have a different archetype. So there's constraints also, very interesting. You mentioned also that straight lines in this theory depend on some kind of, this I'm going to talk like in math, on a Euclidean decline of performance from the archetype. But if you have a more general function, you can have curved triangles and even more weird shapes if it's like L1 norms or stuff like that. So there's some requirements for this to work. But in general, if it's a decreasing function, if performance decreases with distance from the archetype, you'll get shapes that look like triangles tetrahedrons, maybe a little warped. In our experience, triangles tetrahedrons work really well for biological data. All right, so there was another question. Engineers use Pareto optimality like this. They know the performances, so they know, like for cars, I want fuel economy and speed. And then you take all the motors, and if there's a motor that's better than other motor on both traits, they can erase all of these and get the Pareto front. And then, depending on where you want to be, this might be a family car, good fuel economy. This might be a sports car, right? But this is Pareto front. This is what engineers care about. Here it's the same idea, but we don't know what the performances are. Instead, what we have is measurements. We have the car length, the piston diameter. That's what we have. And we infer the number of tasks. So in the space of traits, the family car is here, and the sports car here, and all the cars are here. But if it was a triangle, there's 3 tasks, right? So that's why it's called inverse Pareto. It's an inverse problem. You look at the traits and you infer how many tasks there are. And you want to say that you can, if you don't get it, understand right now, you can look at the lecture notes. I'm just going to continue to immortal animals. Immortal animals. I wrote a song. I wrote again with my daughters. The credit goes to my daughters. The blame goes to me. So does anybody know some immortal animals, for example? Jellyfish, what else? Lobsters, what else?
Eva cells?
Well, you can tell by the way that I use my leaves that I'm a tree that lives 1,000 years. And check out my girlfriend and watch her swish. She's an immortal jellyfish. Go ahead, you can cry. You cry your tears. You humans only live for 80 years. You can teach an aging class, but that won't make you really last, right? Whether you regenerate, whether you degenerate, you're staying alive, staying alive. Whether you're symmetric or your telomeres are hectic, you are staying alive, staying alive. Ha, 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 staying alive. All right, yeah. Okay, that's my song about immortality. Music by the immortal Bee Gees. So of course, immortality doesn't mean these animals can't be killed by predators or accidents. It's just proof the biological material has no physical barriers to last for thousands of years. Put it that way. Cells, eukaryotic cells, etc. And there's 4 different strategies I counted that these organisms achieve this long life. This is a jellyfish. Bats are not immortal. They only live 40 years. We don't count them immortal organisms. Not dogs, not elephants.
Who decides?
Who decides? Good question. So in general, what we want to know is we want animals where this is age and this is risk of death or hazard, right? So you remember that in humans it looks like this. We want animals, actually it looks like this, right? We want animals where it looks like this. And the lower this is, the better. That's to say, the risk of death does not decrease with age. Also, they don't senesce, they don't seem to decline. Their fertility doesn't go down with age. Their agility, their speed doesn't go down with age. That's what we're looking for. That's who decides. The problem is data is very scarce, but there are some experiments that are quite convincing in my opinion. So I want to tell you about those, okay? So there's 4 strategies for immortality. Okay, so one is we call it regeneration. And it's exemplified by 2 critters, the hydra, which is 1 centimeter aquatic creature, and the planaria flatworm, which really has googly eyes, which is I think it's a millimeter. And the planaria is immortal under the knife. What does that mean? If I cut it here and here, it's going to grow into 3 complete worms. And the way that it reproduces, mostly asexually, it cuts itself down the middle itself and 2 worms. Sorry if this is disgusting for you, but how do they do that? Their body is full of homogeneously distributed stem cells. Each of these stem cells can regenerate the organism, and they have telomerase which means that the protective caps around their DNA called telomeres are kind of regenerated. So they can, otherwise, those telomeres with each division, they go shorter, shorter until replicative, you get senescent cells. That's probably an anti-cancer mechanism. And they don't have a problem with that. By the way, talking about cancer, there's something called Peto's paradox. How do whales not die of cancer in 1 year? Because the number of cells they have. And this is getting to the molecular nature of these very long-lived. They have special anti-cancer adaptations that I want to talk about. So regeneration. Also the hydra, the way it works is about twice per month, it does this and this. And it regenerates all the cells every 7 days. And it also has long telomerase. So this is like replacing and replacing, replacing, replacing. Okay, that's one way. And hydra, there's a good experiment tracking 3 million hydra years, which means thousands of hydra across 40 years. And they have a constant immortality, they have a constant death rate like this, which means that 5% will be alive after 500 to 3,000 years. Okay, so that's like very, very low. Even though in nature, they live in the lake, and every season, 99% die every year. But there might still be a niche where the genetic continuation is so important. So this again, there were some mathematical theories in the 60s that says that it's impossible to get something like this because at a certain age, the probability of extrinsic mortality means that the old ones are less evolutionarily important than the young ones, the status of selection. And that theory said that hazard has to increase in all living creatures. And this experiment by Voppel is kind of a proof that this is not universal. There are creatures which, as far as we can tell, the rate of the hazard does not increase with age. And so what's going wrong with that mathematical proof? Check out Appendix 1 in the lecture notes. Yeah.
Do they have like central nervous system?
Yeah, they have a central nervous, not central. They're very simple construction. They have 2 layers, and they have a nervous system, but it's not central. It's like a network.
Do they have memory? Like after you cut them, like do they?
So the question, do they have memory? I don't know, but I think the planaria lab in Stanford that I visited, they learn. I think they can remember across the surgeon's knife. I think so, but can find out. Yeah. Of course, you're asking because, like, if we do that to ourselves and regenerate our brain and we forget everything, right?
Yeah.
Is it worth it?
I don't think so.
So if this is the way that they reproduce, so essentially are they all genetically?
Yeah, they are all genetically identical. They're called a genet. But once in a blue moon, they have sexual reproduction. Because otherwise, by the way, you have what's called Muller's ratchet. If you don't have sexual reproduction, you start getting mutations. And after, 1,000 generations, you can't live anymore. So you have to have sexual reproduction to dilute out the negative mutations. And there are species that don't have sexual reproduction, but they tend to go extinct quite quickly. But these species are very rare sexual. And also, I think the sexual ones, sometimes sex equals death, as you know from vampire movies. But I think they also sort of, the sexual ones have a constant hazard. So, okay, I'm going to continue. So, immortal creatures. The second one is go back to being a baby. So this is what's called the immortal jellyfish, which has a Latin name. These are also very small marine creatures. And their life cycle, they can revert to a polyp that then starts growing on a piece of coral and then becomes a baby jellyfish. Do you remember I told you that our fertilized eggs are immortal because otherwise a 40-year-old mom, 40-year-old dad make an egg, the egg is refreshed, the chromatin and marks are removed and protein damage is somehow erased, and that's how we can continue. Because if each baby was a little bit older, inviable. So there's something about the fertilized egg we have to understand, right? And I'll talk in one of the forthcoming lecture about interventions for aging that try to reproduce what's happening by partial reprogramming in the egg. But this one organ does it. It goes back to embryo and regrows. I don't know what happens to the memories. All right. And then what's another strategy? Compartmentalization. It's also a great psychological trick. So this is the mighty sequoia. And here's a tourist taking an iPhone picture. They can live, somewhere several thousand years, right? Order 10,000 years. Not the sequoia, but there's other pine trees. So there are ones in the Bible, right? They saw the Bible. Maybe even ancient Egypt, pyramid time, which was 6,000 years ago, probably. That's pretty long life, right? For some of these critters, the risk of death actually drops with age. Shocking. Because their extrinsic mortality is lower the bigger they are. It's like they're walking in this direction. They grow and grow. What does this mean, compartmentalization? So the plants, they have a big disadvantage. They can't move. Let's be honest. But they're so good because they're in one place. I cracked you up. They're in one place, and they have to deal with it, right? So they're so smart. They're flexible, by the way. They're strong and flexible. We can learn from the trees. When they have damage or viral infection or bacterial infection, they know how to compartmentalize it. That's to say, to surround it by dead material. We have fibrosis that does something similar. But so when they have damaged tissue, they can sequester it or hide it or compartmentalize it. So, the trees have this ring of moist living stuff and a lot of dead things in the middle. That's why we have tree rings. So the damaged tissue can stay inside and they basically can regenerate. Compartmentalization, right? There's the trees. So funny. That's why my song began, I'm a tree, right? Trying to take a point of view of a tree. Yeah.
But if you would just continue growing bigger and bigger, at some point it's not good for you. I don't know. If you're in the size of Earth, then there isn't any place to grow. So they just keep on growing forever.
There's mechanical limits. You can actually calculate. And there's going to be hand of God. And there's going to be...
What you're trying to say that they don't age, not that they don't die.
That's it.
Okay.
They will die because of accident, extrinsic mortality.
Okay.
But what I'm saying is it's proof that biological material can last for thousands of years. Very, very important point. All right. And the fourth strategy I managed to compile for you, just for you, is dilute the damage by constant growth. So organisms that take that strategy are like E. coli, which is a single cell bacterium. It's cigar shaped and it grows to twice its size by synthesizing new cell wall here in the middle. And then it splits down the middle into two. So, if you have production of houses, you restart. Right? If here you have two houses, you have one. So if we write the SR model as houses minus removal plus noise, we can now write an equation for the houses. Before, we had houses just being made. We had houses growing linearly with time because we don't dilute our altered stem cells when we stop growing. Actually, you can say aging starts when we stop growing. You can say. Before that, it's development. But they never stop growing. I mean, it's two cells, but if we sum up the mass of all the cells, it's exponentially growing mass. Right? Of course, you can do a chemostat where you flow in bacteria, stir it well, and take out liquid, and reach a constant concentration, determined by the way, same growth rate as the fluid you flow in. And that'll continue. Of course, there'll be mutation and stuff, but indefinitely. So we have to add here a dilution term. And that dilution term, the dilution rate is the growth rate. Why? Because when I double my biomass, I half the concentration of houses. And when you do that, you see the houses reach a steady state. They don't grow like this. They reach a steady state, which is their production divided by their removal. And if the houses are a steady state, then they produce some garbage, the trucks clear it, and you reach a steady state garbage. And now you have more and more generations. But instead of garbage going like this, you solve the problem of the growing village because you keep expanding the land, like you're conquering the countries nearby. Whatever. It's a mythical idea. So, right? And so you keep the amount of garbage constant. And now, what happens? This is age. This is garbage. So instead of garbage going like this, it's like this. Of course, there's noise. This is X critical. Are you ever going to die?
Maybe.
How?
Noise.
By a fluctuation. So there is a constant rate of death. This rate of death, it's exponentially unlikely. So if it's X steady state, you remember it's minus X critical minus X steady state divided by the noise. So this is like unlikely, and the higher this X steady state is, the more likely it is. Now growth rate affects this. You see, the faster the growth rate, the less the houses because you dilute them more. So if you go slower and slower, you dilute them less. So as you grow slower and slower, this damage rises, and the death rate should go up exponentially, meaning that there's a slowest growth rate possible before you die. But it's more complicated than that. Maybe the rate of building houses also depends on the growth rate because your metabolism is faster. Or maybe so there's a lot of predictions here, and in principle you can measure this because you can look at individual cells, measure their death rate. That's how you can dilute it out. But there was a surprise in this field because it turns out that this is what's called symmetric division, right? Two cells are symmetric. But it turns out that they're not really symmetric because E. coli has poles. And when a cell divides, you get one old pole and one new pole. This one gets an old pole and a new pole. And when this is going to divide, one of the four daughters is going to get this very old pole. And this one will have, okay? So this pole never goes away because material is already synthesized in the middle. And so there's always, in this exponentially growing culture, one bacteria with a very, very, very old pole. And it turns out when you observe them, that bacteria dies more because the pole accumulates garbage. So there is if you just look at the lineage of that old pole, it does have a rising death rate. So that was surprising finding. Ariel Linder, 2010s.
So it's not true. So the fourth point is not true.
It is true for most of the population.
But like you can also say that like kids are just the extension of like
Do you have kids?
No.
So. Don't get me started! Just kidding. Okay. By the way, in America, you're not supposed to ask questions like that from your students. So I want to edit that out. I don't want to be canceled. But in Israel, it's okay, right?
Right.
Do you have any option to say no? No. All right. Now look. So it's complicated, right? Is it immortal? Is it not immortal? What is the meaning of an identity of a self? Okay. I agree with you. Okay, so 4 strategies for immortality. By the way, there are animals that keep growing, like some reptiles and fish. They'll grow indefinitely. You have to increase the aquarium all the time. So that also, I think, has to do with possibly a dilution strategy in vertebrates. But I don't know the data. Now, I want to tell you something cool. We have.
It doesn't also duplicate the seven. Like if you have gene mutation, then you duplicate the genome.
You definitely duplicate mutations.
So it's not like they have one house here and one house here.
Yeah, but the thing is, the DNA mutations are not the houses. We know that.
Why? You said it's one.
It's because, well, I'll tell you soon. But other stuff, cytoplasmic stuff, gets diluted. But mutations don't get diluted. That's why it's true if you don't have sexual reproduction, after a while, you're going to get so many mutations, you're going to die. Do bacteria have sexual reproduction? Does anybody know?
They have horizontal gene.
Yeah, they have a horizontal gene transfer, so they can get DNA from other bacteria, refresh themselves. Otherwise, we wouldn't see E. coli, which is billions of years old, lineage from the first bacterium. It's still here. Some people say that they're the most important organism on Earth, and they even evolved ingenious containers to move them around.
you.
Me, yeah. Okay. All right. Let's continue. E. coli is very nice. I think it's a really good model organism for asking questions because you can measure so many things that you can't measure in others. So I want to tell you about most of E. coli actually is not dividing. Most of the time it's starving. Because it's out of the body, it's starving, and it's dying, just like us. If you starve E. coli, it'll die. Just like us. The question is, like us, does it have Gompert's law like us? What's going on, right? When you starve E. coli. So it's not exactly aging, it's starving. So Ifan Yang, Ariel Linder built this cool microfluidic device. Basically a lab on the 1 micron scale of the, and you could follow the E. coli dividing, and then here's some food. You stop the food, you stop dividing, and you put in some dye that only goes in when they have a hole in the membrane. It goes into the DNA and becomes fluorescent. So you can now see with fluorescence when they die. And the risk of death when you're starving goes up like Gompert's law. Great. Now, is this really like aging in the sense of, does it have the same damage production and removal like we were talking about in mice? So to do that, Ifan Yang now looked at this signal of fluorescence before they die and telling you something like how leaky is the membrane. Right, the rate of entry of the dye is how leaky is the membrane, which is a good proxy for damage because it's a lot of times the bacteria die because they have a hole, then they reach equilibrium with the environment, they're dead. And it turns out, so let's call that X, that you can measure, without killing the cells, from hundreds of cells, you can measure the dynamics. And when they cross a threshold, they die, because that threshold is, once they cross a threshold, that's how you define death, basically. Like, their whole DNA is fluorescent. And now you can take this stochastic data, just like I told you with the mice, and deduce what's the equation, right? You can do that mathematically. And the equation that best fits this is almost identical to the SR model we're talking about. The only difference is in the SR model, it's a different saturating function. What am I talking about? Here you have bacteria starving, and you deduce really unbiased way their equation, and you take mice senescent cells, and you deduce their equation, and you get almost the same thing. So this is like universality of the equations of damage. This is Nature Communications, if you want to look at it. So this could be a model organism for studying questions about aging, if you want to study the equations. The SR model has parameters. Are they universal between organisms? That's Nave's project, right? MSc project. Nave, Raz. And it turns out that there's like regularities. Like, this is the house production. So the rate of making houses is what most distinguishes mice from men. The long-lived organisms make the houses slower. How do they do that? So I want to get into the molecular mechanism. When you compare whales with bats, naked mole rat, mice, shrews, cows, dogs, you see that this is their lifespan of different organisms. So this is their fidelity or error correction. So first thing, DNA repair, and especially double-stranded break, go up. Translational fidelity, which means how many errors the ribosome make, go up. And protein lifespan. So there are some things that characterize different species with different lifespans. There's trends like that. And this repair, this is not the trucks like in mammals that remove senescent cells. This is the stuff that when you have damaged cells, prevents them from becoming senescent, you know? So this is like in the houses. That's why it makes sense that the house production rate, so humans like have 100 times better double-stranded break repair than mice. They're almost the world record. The world record is whales, bowhead whales. So it's really very striking, and you can understand the double-stranded break repair. Each creature has its own way to increase double-stranded break repair, but it's like right there at the core of. They also have less mutations. If you look at mutations in neurons, for example, that aren't dividing, they rise with age, reach about 3,000 at age 80. If you look at a mouse, they reach 3,000 at age 2. That also has to do with DNA repair, because those mutations, a lot of them are, DNA gets damaged, and the repair introduces mutations. So it's like natural selection pushes the accuracy of these information preserving processes so that you make it to reproductive age. You don't get cancer before reproductive age. And you can stand the damage or injury until reproductive age. And then the way evolutionary theory works is that repair capacity is what you're riding on to reach longevity in optimal conditions in the zoo or in the Weizmann Institute. I just want to finish. Look, so the molecular commonalities I said are reduced IGF-1 function and mTOR, which means the shift to slow growth and big repair, better repair, not just because of that, better intrinsic repair, the enzymes, the regulation, a lot of things. There's also more resistance to damage, like oxidative damage with lifespan. So what do I mean? If you look at the kinds of fat molecules, lipids they make, they're shifted towards lipids that are more resistant to oxidative stress. If you look at the kind of proteins they make, the amino acids like methionine and cysteine that can be oxidized, you have less of them, the more you're long-lived organism. You have less of those amino acid concentrations in the cell. A lot of things together telling us that they change their composition, so they're more resistant to oxidative damage, for example. So you have all these clues. The next lecture we're going to bring together. Okay, they have. So it's a very profitable field to look at, gene expression, and all the references are in the lecture notes. You know what? I think it's a good time now to Mortal Organism, Four Strategies, SR model, mouse evolution, starving E. coli, universality. So maybe somebody who hasn't asked a question yet. I want to hear from you. Yeah.
So about the E. coli, why is it different than yeast? They also divide, and they continue dividing. So why do yeast age and E. coli?
Good question. So why do yeast age? Well, yeast will be star of our next lecture. Yeast age asymmetrically. They bud, at least budding yeast. Fission yeast is like this, same story. Budding yeast is like this. And the daughter passes the garbage back to the mother. And you get a fresh daughter each time.
But it's same as the poles.
It's like the poles, right? Except poles are very, very small. Very old poles are very rare. Here the mother is like, it's also very rare. In an exponentially growing culture, it's like, it makes 30 daughters and they make daughters. So it's exponentially rare, the month, but they're full of damage. That's asymmetric division. So interesting, yeah. You want to ask a question? I want you to come after the class. I want to hear from people who haven't asked yet. And not that your questions are interesting. I don't want to offend anyone. I just want to hear from more people. Yeah.
When we discuss the damage in the E. coli, it's the damage inside a single cell. But in the human model, was it the biomass damage? Was it damage for all the tissue?
In the human model, remember our candidate for X is something like total senescent cell load, including the inflammatory signals they secrete all over your body. So it's not in the cell, but the houses are inside the cell. Does that make sense? Or did I answer your question? Is that what you wanted to ask? Yeah.
Does having babies make you age faster in humans?
Does having babies make you age faster?
Also, like
So
the
is
number of
yeah. So there's this one of these lovely plots like this, where the hazard ratio is number of completed pregnancies. I don't remember, between 2 and 3 is the lowest. Am I right? Yeah. So it's like Ido is a gynecologist is doing a sabbatical now with us. So, what else?
So like the hazard ratio is higher below 2.
Yeah. It's like healthy to have a couple of, what is it, pregnancies or completed pregnancies? Completed pregnancies. What?
It's correlation, it's not causality.
Correlation, it's not causation. It's a correlation for sure. Because if you're very sick, probably you won't be able to have a pregnancy. So I don't know actually about that side. Yeah, there's all the social stuff that goes, like human stuff. But I think in mice you can also probably measure that. I wonder what happens. What about you? What's your question? Yeah. You? Yep. Yeah. You? Your?
What happens in like close like relative of ours, like Neanderthals and like chimps? I've like you said it.
You say
Very
close relatives to humans like chimps. Yeah.
like a Neanderthal, like
Neanderthal.
yeah, most like I don't know, like
The
do
closest
we have
relatives.
like a big difference in our longevity compared to primates who are close to us?
Okay,
Or
yeah.
is it being
So primates, I know a little. Primates, we're the longest lived primates, and I speculate that's why it's so hard to push our longevity because I think we're optimized for slow development in the primate chassis. Just yeah. But the other primates, so chimpanzees, for example, they can live to let's say 50. And another interesting thing, they can have birth till 50. They're fertile till 50. They're fertile to the end. So in humans, we're also fertile. I mean, till 50, but there's extra lifespan that you don't see in chimps. And as I said in the first lecture, there's theories about that where the evolutionary theory is that you gain benefit to other organisms that share your genes, like your kids and that's called kin selection, by sticking around in different ways. So grandmother hypothesis and other ways. Hard to prove. There's another hypothesis that animals where the baby is very big compared to the mom, like humans and also C. elegans, the mom needs to be so robust in order to give birth that it has extra repair and resilience, and that's why you have long life. So that's another theory by Kathleen. There's some evidence for that. So this is very hard to prove, but
what about Neanderthals?
Neanderthals, I don't know about Neanderthals. I do know that Stone Age skeletons, 5% exceeded age 65 according to the bones.
Wow.
But 50% of deaths were from violence, on the other hand. So, all right. What shall I wish you? Long, happy life with quiet nights, pets if you want them.